So, well, regardless, I'll maybe need to hand wave or something. But they divide through binary fission, which is to say that when they make their chromosomes, recall they synthesize their chromosome and duplicate that thing, and they attach it then to the cell membranes. So when it divides, it's kind of pulling the chromosome apart on either side. Oh well, sorry about that. So that's pretty straightforward because it's they're simply making two daughter cells. Now we know bacteria are haploid, okay? So, but some of these other ideas like, diploids, that's what we were considering when we looked at humans. We know many organisms such as plants can be polyploid, multiple copies of their genomes. So because certain organisms can be diploid, that allows them to do kind of different kinds of reproduction than simple, binary fission. So if we consider eukaryotes, they are able to do mitosis, which is pretty much, it's a similar process to binary fission in bacteria in that they're simply making two identical daughter cells. So in that process, in kind of the idea of cell division, what they'll do is they'll take their chromosome. Okay, so we'll have a long linear chromosome. It will now duplicate that. So it takes the one strand of DNA and duplicates that, and then now you create your chromosome with the two homologous chromatids, the two sister chromatids, I should say. So we have our sister chromatids, and in mitosis, in the various stages, so interphase is where the DNA synthesis occurs. Then once it goes into the actual division phase, the first step, or the four stages of that, of course, prophase, metaphase, anaphase, and telophase. Excellent, thank you very much. So we know that at the beginning of prophase, or in mitosis, the chromosomes will simply, you'll have some cell, and the chromosomes will simply be lining up in the center of the cell, the two homologous chromosomes. And then they will be pulled to either side of the cell. Okay? So that's mitosis, and where you're simply creating two identical kind of clonal daughter cells. One thing that makes diploids and polyploid organisms different is the fact that they can also do meiosis. And meiosis is only possible because of diploid genomes. So here there are not just one cell division, there are actually two stages to the cell division. So in meiosis, again, we have the homologous chromosomes in there, but the difference in...
Did you just start?
I just started.
Okay.
And mitosis, they all line up in the sister chromatids part. The difference in meiosis is that the two homologous chromosomes pair up together in the center of the cell. Okay? And so, but the difference is, in the book, recall, it presented the homologous chromosomes like pairing up kind of like this. Where one of the sister chromatids on either side would match up. That's not actually the way it works. Rather, the two sister chromatids come together, one on top of each other, so that you can have crossing over in either of the arms. So, actually, these things are now paired now. Like I had drawn before, but backwards. But the idea here is that because these are diploid organisms, you have the two copies of every chromosome. You can have this crossing over, homologous recombination, which now provides for genetic diversity. We can shuffle the alleles that are present on those various, between the chromosomes, making new combinations of alleles. Okay. And then in I'll come back to that idea in a moment, but the idea with, after continuing on in meiosis, after that crossing over has occurred, then what we're going to see is each of the daughter cells will get an equal number of the chromosomes, but then in the second stage of the division, then these chromosomes will be split just like in mitosis. where all of them will be lined up in the center of the cell, and the two sister chromatids will be pulled apart from one another. Okay? So meiosis phase 1 is kind of unique to itself, but meiosis phase 2 behaves just like mitosis. Sort of, except in the end, the products of mitosis, you still get two diploid cells, whereas in the products of meiosis, at first you get two diploid cells, but then that goes further to 4 haploid cells. So what is it, what's the idea behind this crossing over? So I'll come back to this when we look at the recombination a bit more. But again, the purpose here is that, say we look at some big chromosome or whatever, and maybe let's draw another one. Okay, so we have like an A allele, B, C, A, B, C. Okay. Well, if there were no crossing over, the daughter cells would always get exactly the same alleles. There would never be any differences. So just like I'd been talking about the other day in population genetics, there would never really be any heterozygotes for anything, because you would always just get, well, maybe not quite, but the offspring would always just get, dominant A, B, C all together. But the difference with the crossing over is because we can have DNA being exchanged between these arms. You can create new combinations of these chromosomes, or maybe now it's making some chromosome that is, let's just draw one of the arms here, that is instead maybe like A, I think B, C or something. And so by scrambling the alleles in the chromosomes, this provides for genetic diversity. And this is, again, much of the basis behind sexual reproduction in diploid organisms. We can scramble the chromosomes, make haploid gametes, and then recombine those gametes. So in Mendelian genetics, we were looking at kind of basic situations in which we were looking at how the various genotypes, or the various alleles, would combine in order to create various genotypes. So we learned about dominant and recessive traits. The basic idea, of course, a dominant trait generally expresses itself over a recessive trait. And generally that's because a dominant protein generally is functional, whereas maybe a recessive protein is less functional or even non-functional. But we learned about different kinds of dominance, but generally we considered, we thought about it as just complete dominance. The dominant allele is more or less working protein, the recessive one is more or less non-working protein. Or it does not work, does not show a phenotype if you had a dominant protein present. Okay. So we learned about the single gene crosses. That is, we considered cases like, okay, we only crossing a homozygote dominant to a homozygous recessive, and we would see how we could make heterozygotes from that, and or we could say make, this would create 4 different heterozygotes. But the idea here was these Punnett squares of that sort of thing, where we had very simple Punnett squares in order to figure out the different combinations of gametes that are produced in order to provide for then the different possibilities of genotypes that could be made in the offspring. We also looked then at, we saw how we could do these crosses over different generations. We could take, say, a parental generation, cross those to produce like an F1. We could take an F1 generation self-cross, create an F2, because oftentimes the traits, like in here, if you do this, we'd see that, okay, we see homozygous traits in a parental generation. We see those homozygous traits disappearing in a F1 generation, but then they might reappear in the F2, because if we self-cross these guys, that would go to A, some of those, 1 to 2 to 1, something like that. So that's kind of just the basic idea behind these different generations, in that sometimes when you cross certain alleles, those phenotypes might disappear in one generation and then reappear in the next. So in this case, like the recessive phenotype would disappear but then reappear subsequently. We also considered 2 gene crosses, that is dihybrid crosses. So the idea here was we considered maybe AABB, crossing that to, say, recessive AABB. Okay? So there we needed to set up much larger Punnett squares in which we considered the various possibilities of the gametes that are produced. These gametes produced as a result of kind of mixing due to crossing over and such. Now here it's pretty straightforward because they are all the same alleles, but recall that we made Punnett squares that are 4 by 4 that are produced, actually maybe this other way, that would make more sense, that presented the different possibilities of gametes that are produced. Okay. So in this case we are presenting the 4 different kinds of gametes that could be produced here, and we would make a Punnett square comparing that against, I guess in this case the 4 different kinds of gametes are all the exact same thing. But this is what allowed us to create those 16 different possibilities of genotypes that could be produced by mixing any of these gametes. Okay? So then in kind of the traditional Mendelian genetics, we saw that this produces a phenotype, or there, with the 16 different phenotypes, this produces genotypes that correspond to 9 individuals that show the dominant, 3 individuals that show, or I'm sorry, both of them being dominant, 3 individuals that show one of those dominant traits and recessive for the other, 3 that show one of those recessive traits but dominant for the other, vice versa. And then there was the 1 individual who showed the recessive characteristics for both traits. So that is to say, if you do these 2 gene crosses, starting out with maybe homozygous on either side, we tend to see these 9 to 3 to 3 to 1 type phenotype or genotype ratios being produced. But these correspond to 4 different kinds of actual phenotype. So one thing that we were assuming whenever we did these Mendelian genetics was the idea of independent assortment. That is, that 2 alleles that came from 2 different genes behave differently from one another and are not, will always show up in, I should say, can't produce all the different possible forms of gametes. They're not going to stick with one another when they're forming gametes. The reason why is we assume that, say, A and B were always present on either different chromosomes, and since we know the chromosomes align independently during mitosis, you can always end up with gametes that have various combinations of those. But we also know the idea of independent assortment is that even if you have 2 genes that are still present on the same chromosome, so A and B are still present on the same chromosome, they're far enough away such that a crossing over event most likely occurred between them, so it could still scramble the possible combinations. So we can, in fact, still see gametes that are, AB or A recessive B, etc. When we got to eventually this point. Okay. This is opposed then to the idea of linkage. So linkage simply means that if we were to, say, have heterozygotes A and B, this was because perhaps we had a chromosome where perhaps for the two homologous chromosomes, one of those chromosome arms had the dominant A and B, and the other had recessive A and B. If these genes are linked, that is, they will stick together throughout the meiosis process because there cannot be a crossing over, or it's very rare for a crossing over event to occur between these. What we would see is that you never saw the 4 different kinds of gametes. We would actually only see, if these were completely linked, we would only ever see 2 different kinds of gametes. Okay? So this was the idea behind linkage, in that rather than seeing, more or less a random distribution of the 25%, 25%, chance of seeing all of these gametes, you see some skewed ratio where, I guess it's completely skewed and these are completely linked. You only see these 2. So just the idea of linkage is that you have genes close to one another on the same chromosome, which slows down or reduces the chance of crossing over between them. So the genes are more likely to stay together when producing gametes. So another thing we were learning about was genes involved in sex determination and the genes that are present on the X and Y chromosomes. I guess here more we focused on genes present on the X chromosome. So an important thing to remember there is that generally genes that are if you have alleles for genes that are present on the X chromosome, so like a dominant or recessive, we tended to see the fact that oftentimes males would show a recessive phenotype, whereas females would not. And that is, you would see these sex differences in the phenotypes we might see. The reason for that is because oftentimes males would get just their one X from the mother. Maybe the male would inherit the recessive gene from the mother. The mother was a heterozygote, didn't have that genotype. Whereas, say, daughters, they would have to get the X from the father, who probably didn't have that phenotype either. It was dominant. Okay. So just the idea of the sex linking just meant that we would see that even though the genotypes were always kind of working out the same, that males tended to show certain phenotypes which would be different from female offspring, simply because the males would only get the one X, and so since they're kind of effectively haploid for that chromosome, that one chromosome, they only needed the one copy of that recessive allele. Okay. So epistasis. The idea behind epistasis is that, up till now, we had assumed that genes A and B were creating proteins that were functionally independent of one another. That is, if A is recessive, that has absolutely no effect on the activity of B. Or if B is recessive, that has absolutely no effect on the activity and the resulting phenotype of A. The idea of epistasis, on the other hand, is assuming that functionally A and B are connected. So if A is mutated somehow, or A becomes recessive, that's going to have some sort of impact on B, and vice versa. Okay? Or a good example of this was like the idea of gene duplications, where maybe A and B are the exact same thing. You get rid of B, well, you still see the dominant phenotype. There is no special phenotype right here because it always looks like AB together. You knock out A, it still always looks like AB together. It's only if you got rid of both of them do you actually see a recessive phenotype. Okay? So the key concept behind epistasis is that because genes are functionally connected somehow, we see a variation in the ratios that we observe, or the phenotype and genotype ratios we observe in the offspring. That is to say, in a normal Mendelian case where A and B are functionally separate from one another, we see these 4 different phenotypes, and each phenotype was being produced by 9 genotypes, 3 genotypes, 3 genotypes, 1. In epistasis, we might still see 4 or less phenotypes, but the ratios that are, the genotype ratios that are producing these phenotypes are altered. Okay? So in the case where I was mentioning that maybe A and B can substitute for one another, we would only see 2 different phenotypes, and the dominant phenotype is supported by 15 different genotypes, whereas the recessives only by 1. Okay? So it's these altered, you don't need to know what the ratios are, but it's the idea that there are these altered ratios that we will observe in the offspring, or the possible genotypes making those phenotypes that supports the idea of epistasis. Gene redundancy, I had just been mentioning that, how you can have different proteins that are able to, you knock out one gene and it has no effect on the phenotype. You don't see any special recessive phenotype appearing for that because another gene is covering its function. Effectively, you need to knock out both versions, or as many versions of that gene as possible to actually see something. So, yeah, as I just mentioned here. Lethal alleles. So we know that certain genes are critical for the cell because if you, at least in a diploid organism, because if you get rid of both versions of that gene, obviously the organism cannot survive. So we actually are never able to see an organism with that particular genotype. So, for example, say and the example of this was the Manx cat in class. Recall where normally cats have this recessive phenotype. Okay. But say a cat might have, it could have a heterozygous phenotype. In that case, this protein actually interfered with their spinal cord development such that they could develop their normal spinal cord down to more or less the beginning of the tail, but then the spinal cord that went into the tail could not develop. Okay, so they were tailless. Though interesting, if you had an offspring, say you had maybe 2 heterozygotes that were mating, we could see phenotype or genotypes produced that are the AA, and we would see heterozygotes, so we would see 1 of those, 2 of those, and presumably, going back to kind of the 1 gene cross, the 1 to 2 to 1 type things. Of course, these 4 possible genotypes can be produced, but in terms of offspring we actually see this one and we would see those 2, but we would actually never ever see that. Why? Because being double dominant in that case is lethal. They'd never be born, so we never actually observe that phenotype. So this is the basic idea behind then the lethal alleles is that we know that an allele is necessary or some certain allele possibility is detrimental for an organism if we actually never see the homozygous situation for that allele. Okay.
Are you sure you don't mean the Manx gene is recessive, sir? Because if it was dominant, wouldn't it?
Or maybe it was recessive. It doesn't really matter. Correcting me is fine. Yeah, maybe it was recessive.
Sorry.
Yeah, thanks for, I can't recall, but it sort of doesn't matter in this situation. Whether it was dominant or recessive. Just the purpose, the point is that I thought it was dominant because the protein is actually interfering with spinal cord development, but this was a case where it was incomplete dominance. Recall, heterozygote was incomplete dominance, that is, the effect was not as strong as in the homozygous situation. Because if it were just a single recessive, they would have no phenotype if it was a normal complete dominance. Well, anyway, it doesn't really matter, but just the idea of lethal alleles. We don't see the homozygous situation for that.
The dominant allele was the dominant allele was the dominant.
Okay, I was right. Thanks. Mitochondrial DNA and chloroplast DNA. We know that these sets of chromosomes are separate from the nuclear chromosome. And as a result, they do not cross over. You know, there's no exchanging of genes between those genomes and the nuclear chromosomes. Okay? Also, we know that those chromosomes are generally inherited from just one of the two parents. So, for example, in humans, we get our mitochondria from the egg cell, from our mother. Okay? The sperm actually do have mitochondria as well, but somehow they're degraded when they actually get to, when the sperm merge with the egg cell. Okay? Plants, like most of them, actually get the chloroplast from the pollen, though it varies. Right? Maternal effects. So we saw this just the other day for development. So the idea behind maternal effects is that it does not matter what genotype the offspring has. The phenotype of the offspring is enforced by the genotype of the mother. Okay, so that sounds a little weird, but recall in class what we had seen was an example of snails, where certain snails, say they had, I guess, a recessive allele or something, and normally that would say, okay, your shell spins clockwise. Okay? So the offspring here is recessive, and the recessive alleles tell it to spin clockwise. But even though the offspring's recessive, the dominant A protein is coming in from the mother. And it's that dominant A protein coming in from the mother that enforces the phenotype associated with the mother's genotype on that offspring. So instead, the offspring actually coiled the other direction. Okay? So that, again, the idea of the maternal effects is the genotype of the mother establishes the phenotype of the offspring. But this will carry over in that, once again, from this organism, it's the genotype, assuming this one was female, it's the genotype of this one that will now carry over to the genotype of, or the phenotype of the next generation. So even if that offspring, say, maybe got another gene that was A, so here's AA, this genotype enforces the phenotype in the next generation again, so now it's going to coil like that, even though it was a heterozygote. So we know that there can be maternal effects if the phenotype and genotype don't always match. And the phenotype we see for one organism actually matches to the genotype of the parent. Okay, so we saw this in development the other day, where it's a lot of the proteins that are coming from the mother cell that are telling the egg, or I should say the oocyte and initial embryo, how to start developing, how to grow, which direction's which. So it's more or less just this influence of the mother's protein. So epigenetics, we had seen more about this later on in the semester, but the basic idea here is, we know that we can turn on and off genes through DNA methylation. You methylate a gene, and this can impact the ability of transcription factors to bind to the DNA. If you heavily methylate the DNA, this can start to recruit other proteins that lead to more or less DNA compaction and completely shutting down of that gene. Now, in terms of how it relates to classical genetics, we never really did any sort of problems that involved gene silencing or epigenetics, simply because if you can just randomly shut off genes here and there, regardless of what the allele is, obviously this completely messes up any sort of prediction of what the genotype is relative to phenotype. Just as we saw with that agouti gene example on the last exam, that it really is irrelevant that the offspring had the dominant allele because it still had the recessive phenotype. Okay, because of the epigenetics. So, but just something to keep in mind, in that due to these other effects, this can disconnect genotype from phenotype as well.
Just something I've been wondering for a while. How exactly is the methylation removed? You said that it's repaired during
recreation. So yeah, when eggs are created, the DNA methylation is almost entirely removed from the genome. And so the way that there are many DNA methyltransferases expressed that will cut the methyl groups off of the cytosines. And the idea behind this is because, as I mentioned, like over our lifetimes we accumulate lots of methylation in our genome, shutting certain genes on and off and such. And presumably sometimes that can turn on cancer, pro-cancer genes. And you don't necessarily want to transfer on some of those effects. So in sperm and egg cells, almost all of the methylation gets wiped out to prevent these sort of maybe detrimental methylation from being passed on.
I thought you said that it was put on by the polymers, or it was put on by something later after it had been copied. Or am I just confused? In an organism, once it starts developing, there are DNA methyltransferases that we know that's how those methyl groups get added onto the DNA. Okay. There are methyltransferases that are expressed that will combine those onto the DNA. But in terms of where they actually add them on, it's kind of random and it's not quite clear why it happens. But if you go back to that exam problem, the key there was why. I can kind of see where you're coming from, because your question more or less is, it's the disconnect between, I had said, well, if the methylation gets wiped out from the egg cell, how could it possibly have been transferred on in that case? The difference was when the egg cells are initially made, the methylation gets wiped out. But remember, in females or animals, females are born with all of the eggs that they will ever produce. And so in that case, because the mother was eating all of these methyl-rich diet, it was even methylating the DNA in the eggs that were already there. And that's why we saw that effect. Okay, now I understand.
Yes.
Thank you, 1 second.
Go ahead.
Write this down.
Yes, but normally any methylation in your DNA is not going to be passed off onto your offspring. So bacterial genome packaging. We know that DNA is pretty big and relative to the size of the cell. So it needs to be compacted somehow so it actually can fit inside of the cell. So how do cells do this? So for bacterial cells, most bacterial cells have circular chromosomes, which even then, a complete circular chromosome is a lot bigger than the size, if you were to take it as a whole circle, it's a lot bigger than the size of the whole bacterial cell. So the way that bacteria compact their chromosomes is they more or less put it into loops like this. It's kind of like if you were to take a rubber band and grab it at various points and pull it to the center. Necessarily, it's going to get a lot smaller. Okay? But then the other thing that they do is they take these loops and they start twisting them. So bacteria supercoil their DNA. And again, if you can imagine a rubber band, you take a rubber band and you twist it, what's going to happen? First off, it becomes linear, it becomes straight, but if you relax it a little bit, it'll crumple up on itself and form a very tight ball. Exact same thing here. It twists the DNA, then when it relaxes, it collapses into this really tight ball. So it's able to compact something that was originally some really big loop into something really tiny. Okay? And that's how bacteria are able to fit their circular chromosome into the complete cell. Now, eukaryotes need to do something different. So we know in eukaryotes they do have, well, in the mitochondria and chloroplast of eukaryotes, they do this sort of thing because those genomes look like bacterial chromosomes. But the eukaryotic chromosomes present in the nucleus, we know are linear. So the way those are compacted instead is we start out with some really long linear chromosome. Then, in the first stage, we take that DNA and we wrap it around proteins called histones. That's a pretty bad picture. Okay, let's try that again. We have, say, some histone protein, or histone proteins. We wrap DNA around it. Whatever. So we wrap DNA around it, and by doing that, it's like kind of
wrapping string around some sort of bead or something. Okay, we've taken something longer and made it somewhat shorter. Now, the nucleosomes, now the idea is it's kind of like beads on a string, in that you have a series of these. There are these little gaps and linker regions in which we have regions of the DNA wrapped around these histone complexes, a little linker, and then it's wrapped around more histones. Okay? So the chromatin. And then the idea of the 30 nanometer fiber. So we take these sort of beads on a string and now wrap that around each other. So we'll take all of these things and start wrapping those into some sort of helical coil. And you can kind of imagine how it swings around like that. And by doing this, again, it's compacting the DNA even further. You know, the length of the DNA molecule stays the same, but the difference is rather than it being stretched out really long, you know, by twisting it, we've kind of made use of three-dimensional space, you know, and we've compacted the actual volume to something much smaller, or rather we've made just more efficient use of the volume. Okay. So, and then these 30 nanometer fibers can be further compacted. So inside of the nucleus, there are these nuclear matrix fibers. And these 30 nanometer fibers can be attached to these things and loop around them. Similar to what the bacteria were doing. You know, they'll take these long things and loop them. And this is now the basis of the entire chromosome. So recall, if you had a linear eukaryotic chromosome, it's actually just a series of all of these loops of 30 nanometer fibers going like that. Okay, so that, recall, I had shown a picture where we had looked at a duplicated chromosome, so it had the two sister chromatids, but you could see all these loops coming out of that. You know, that was the basis of what this was, these 30 nanometer fibers that are connected to central proteins in the chromosome. So in this manner, this is how eukaryotes are able to fit all of their DNA inside of the nucleus. So some of the types of DNA, well, we had seen that, or just in terms of the structure of DNA, we had seen that DNA can potentially take different forms, like aDNA, bDNA, zDNA. Really the only important one, biologically important one, was the bDNA, which is the normal twisty form that we see in cartoons. aDNA, or the A form, was more relevant to the structure of RNA molecules. Let's see this. But yeah, euchromatin. So we know that euchromatin is less densely packed regions of the chromosomes. And generally it's because they are transcriptionally active. That is, the DNA is not as densely bound in these 30 nanometer fibers. The DNA is not as densely compacted onto the histones. And therefore it's much more available to be read by RNA polymerase and bound by transcription factors, things like that. Okay. Heterochromatin, on the other hand, is DNA that is very tightly wrapped. So for example, we saw that for most chromosomes, the heterochromatin tends to be found especially at the centromere and the telomeres. And for example, also like a good example of this is like the bar body, you know, the deactivated X chromosome that's present in females because they have two X chromosomes, one needs to get shut off. One of those X chromosomes is entirely converted into heterochromatin and then just kind of dumped off to the side of the nucleus. And indeed, that's the fate of all heterochromatin. It gets kind of pushed to the side of the nucleus where it gets out of the way, whereas all of the euchromatin is in the center of the nucleus and can be transcribed. DNA polymerase. So how does DNA polymerase work? The basic idea holds true for both eukaryotes and bacteria again. And that is that you have some double-stranded DNA, and we have to open this thing up. So we open up the DNA. Then we can have DNA polymerase complexes coming in on either side of these replication forks. Okay, so we open up the DNA, there's going to be a replication fork going this way, replication fork going that way. Now, in the DNA polymerase, the big problem with it in both bacteria and eukaryotes relative to RNA polymerase is that it cannot just start laying down DNA on its own. It needs a primer. Okay. So for DNA synthesis, you need an enzyme called primase that will lay down a short RNA primer. It's something for the DNA polymerase to bind to and start extending off of. So it'll take this RNA primer and start attaching just normal deoxy ribonucleotides to the end of that. Also, we know that as the way the DNA polymerase extends, there's a problem because DNA polymerase extends 5' to 3'. Okay, so in this case, let's say that's 5' here and 3' here. Okay, so it's going to extend 5 to 3. But it has to do the exact same thing in the opposite direction because we have two DNA polymerases that are connected. So while one DNA polymerase can bind to just a single RNA primer and just go from there, the other DNA polymerase, more or less, it has to synthesize in this direction, but it's getting pulled in the opposite direction. Okay, so this is the idea behind the leading and lagging strands. The leading strand is synthesized consistently in just one direction. The lagging strand is what is being made in the strand that's kind of the opposite direction from which way the polymerase is actually moving. So the way the lagging strands are made, or rather the way that the cell compensates for this, the fact that the polymerase is moving one way but needs to synthesize in the opposite direction, is it will take its DNA, sort of loop it around, such that the DNA that is passing through the polymerase is still 5' to 3'. Okay? And we saw some animations in class about that. It's not super important, but just the idea that the DNA, it's still synthesizing 5' to 3', even as it's moving in this direction on the lagging strand. But the lagging strand DNA polymerase can only do this for so long. After a while, it needs to let go. Okay, it can only loop the DNA for, it can't do it forever. So the DNA polymerase on the lagging strand eventually lets go. It has to jump forward to another region of DNA further up, bind to a new RNA primer, and then extend again. Okay? So to kind of demonstrate this, we would have a leading strand that just keeps going, 5' to 3'. A lagging strand would have been synthesized here, and maybe there's some RNA primer right there. Well, a new RNA primer gets laid down, and now the next DNA polymerase has to come in, bind to this, and start extending again. And it sort of varies whether it's bacteria or eukaryotes, what happens when it gets to the other RNA primer. But regardless, eventually that RNA primer is degraded. DNA polymerase fills in the little gap. And recall then there's a nick in the phosphate backbone right here because DNA polymerase cannot connect the phosphate backbone between already existing nucleotides. So then you need DNA ligase to come in and close the gap in the phosphate backbone. And now we have a continuous strand over here. But recall, this is because of these replication forks going either direction from the initial position in the DNA that was opened up. This is happening on both sides. We know that DNA polymerase has pretty good fidelity. That is, it rarely makes mistakes, but sometimes it does make a mistake. And it puts in the incorrect nucleotide. Usually when it has put in the incorrect nucleotide, as we saw in mutations, what happens? The nucleotides do not hydrogen bond correctly, and so they bulge. They kind of push each other away. And DNA polymerase can actually detect, this bulge in the DNA, not initially when it puts the nucleotide in, but after it moves a few positions in the DNA. So we have some little bulge. The polymerase is going here, but it will detect that there's this bulge, and when it catches that, the polymerase stalls. Okay? It stalls. It will physically back up and degrade a random amount of nucleotides around that region. Once it's degraded all those nucleotides, hopefully it got the incorrect nucleotide as well, it will start back up again, and again, because it has pretty good fidelity, most likely this time it will put in the correct nucleotide. Though, this is something I hadn't mentioned, but sometimes DNA polymerase does not catch these errors, or it can get past these incorrect ones. So this is also a reason why mutations can occur. Because yes, DNA polymerase can catch mistakes. It makes mistakes, but it can catch its mistakes. But it's not 100% effective in catching those mistakes. So even then, there are still mutations that arise from just the lack of fidelity of DNA polymerase. Yeah, I said that. Okay, telomeres. So we know a big problem with eukaryotic chromosomes is because they're being linear. The replication of the end of the chromosome is quite problematic. So, and this also ties into the issue that DNA polymerase is unable to bind to the DNA on its own. It needs a primer. So at the very end of a chromosome, there is a 3' overhang. Okay? The reason why this 3' overhang exists is because there is no location for an RNA primer to bind from which DNA polymerase can actually fill in that gap. Okay? So there's this overhang. Well, how do cells deal with this overhang? Because if it didn't deal with this, this overhang would, it would cause the shortening of DNA every time the DNA was replicated. The way that cells are able, eukaryotic cells deal with this overhang and protect the ends of the chromosomes is through the activity of telomerase and special sequences at the end of the chromosomes called telomeres. So what telomerase will do is telomerase has a built-in primer that is able to complement a region of that overhang. And also within the telomere, there are highly repetitive sequences, which also, again, allows the built-in primer to bind to that repetitive sequence. Telomerase will bind to the overhang and then use its built-in primer to extend the strand. Okay? At which point it will let go, it will shift over, use its built-in primer to bind to the end of this again, and extend over and over and over again in these set nucleotide segments. So having done that, we now have a much longer single-stranded overhang. Well, gee, now RNA primase can come in. It can lay down a primer, and just the normal DNA synthesis machinery, normal DNA polymerase, can now extend off of that primer. And we'll still need DNA ligase, to connect here. But notice now, we've extended the length at the end of this chromosome. So we don't need to worry so much about, oh, there was an overhang here, it's getting shorter. Well, no, we've preserved the length of the chromosome. But that RNA primer is degraded, we still have a 3' overhang. There's no way to get rid of that thing because, again, coming back to the issue of the polymerase has no way to fill this in because there's nowhere to bind. So there's always a 3' overhang. All we can do is extend the end, or kind of push that overhang out farther. Though eukaryotic cells do have ways to protect this end, and that is it kind of like twists the DNA around itself. It has a bunch of proteins that are able to bind the telomeres and wrap it around to help prevent degradation. Homologous recombination. So coming back to the idea of crossing over. And so during meiosis, phase 1, specifically prophase 1, when we have the two systems, well, I should say this is true for any molecules of DNA that are exactly the same. So long as you have the homologous recombination enzymes, it will be able to do crossing over. But at least in a biological sense, it's most relevant, during meiosis phase 1. So if you have two molecules of DNA that are pretty much the same, we can have recombination between them, or crossing over between them. So, which is to say, two of those strands get nicked, and then the DNA is able to invade the other strand. So it gets connected here, and it's effectively they get reconnected to the opposite strand on the opposite molecule. When they're reconnected like this holiday junction is able to be moved. So it gets pushed along now, and this blue strand now starts crossing over down here. The red strand starts crossing over up here. So effectively what we're making now is, make sure I get this right. I guess I was pushing that way. Regardless, the point is that we've now exchanged strands in this region. So we're exchanging strands and creating these regions that are mismatched. We have here and here these mismatched regions where most of the nucleotides will be the same. But as we've learned, different chromosomes can have, because there are allelic differences, presumably might have single nucleotide polymorphisms or something like that. So maybe there are a few nucleotides like, here was an A, T, but then maybe down here instead it was like a GC. This is again a source of mutations. Actually, I guess I should see it.
Just write it all in one color. We'll
A,
understand
T,
the
and C.
location.
There we go. There we go. Okay. So, but the idea here is that this can introduce, mismatches. Well, anyway, so we have this holiday junction extending for some certain range in the DNA. Eventually, this thing needs to be resolved. It can happen one of two ways. One way to do it is to cut the two strands that were initially cut, the two strands that are crossing over. If we cut the two strands that were initially cut, they will reconnect such that this connects here and this connects here. So that there is no actual recombination event. That is to say, on either side of this region, blue is still connected to blue, red still connected to red. So that all that we've really done is created mismatches in this region. Okay? Now these mismatches will need to be resolved, and we know that the DNA repair machinery can come in and fix these mismatches. But notice, just like we had on the exam, why are mutations inevitable? Why can they not? Why is it impossible to stop mutations? How does the DNA repair machinery know which one is which?
It doesn't.
It doesn't. Exactly. So it's going to cut either one. If it cuts C here, it'll return it to the normal chromosome. If it cuts the A here, it's now converted it to the nucleotide on the other chromosome, and presumably we've introduced a kind of a mutation. It's not quite a mutation because it's still present on the other chromosome, but imagine there were multiple of these mismatches. We can presumably shuffle the single nucleotide polymorphisms that are present on these chromosomes, creating new combinations of these polymorphisms. But on the other hand, say we do not cut the strands that were initially crossing over. Say we cut the strands on the outside instead. If we cut the strands that were on the outside, that's right. We still have a mismatch region where they had crossed over. But now we actually do have recombination. That is, by cutting the strands on the outside and reconnecting these, we've more or less taken two different chromosome arms, cut them, and completely moved them over. So we've taken the chromosome arm that was present on one homologous chromosome and passed it on to the other homologous chromosome and vice versa. Okay. So again, this was the idea behind why we could shuffle alleles, because, I guess I erased it, but as we saw earlier, you had dominant A on one chromosome. I guess you had dominant A, dominant B, recessive A, recessive B. Now the fact that we have shifted them, we have A and B, so hence why we could produce those different possibilities of gametes, the independent assortment. We say A and B are not linked, because they didn't stay together in this case. I guess they're far enough away. So this is the recombination. The other way to do it, this was the holiday model. The second way was
the holiday model. You called that the holiday junction.
Yeah, holiday junction. And this was the
that's always referred to as the holiday junction, but not this model of recombination.
No, this is the holiday model of recombination, in which we just have a single cut here, and it crosses over. We have the holiday junction, and it goes in just a single direction before being resolved. Because I was going to say, the second model of homologous recombination uses the double-strand break model. It still has holiday junctions.
My mistake.
But,
it's called the double.
Because I was just about to say,
the double-strand break.
Yeah, the double-strand break model now, the way that one works, it doesn't actually, it's not super important how this one works, you don't need to know that. But the basic idea is, rather than nicking one strand on either molecule, rather it cuts the strand on just one of them. Okay? It degrades some of that DNA, and after that, this now relies upon the DNA repair enzymes to fix this. Okay? So in the double-strand break repair enzymes, we'll take the homologous DNA strand, use that as a template to fill this thing in, more or less. I guess they fill that in. Well, anyway, but the key here, something like that. Well, okay, I'm drawing it all wrong. But the point is that in the double-strand break model, there are actually two holiday junctions. Okay, here we had seen one. In this case, there are actually two. Again, I'm drawing it all wrong, but the point is that there's one junction here and a second junction here that goes in both directions. And both of those need to be resolved. Depending on how they're resolved, again, doesn't matter all the mechanism of what's going on, but we can still have either recombination or non-recombination, and we will still see these mismatch regions with potentially mismatched nucleotides that need to be repaired. Okay. But these are the two different possibilities of recombination.